Welcome to part 2 session of BI 110. So we are talking about prokaryotes. Remember in the previous video we talked about just the introduction, the best examples that falls under prokaryotes, and also some characters. Like we mentioned 1 or 2 characters, so it's important that you watch the other video as well, so that you connect what we are trying to say here. All right, without even wasting our time, let's get started. Okay, so what are these prokaryotes? So prokaryotes, they are single-celled organisms that actually are made up of one cell. All right, so they are made up of one cells. So that's what we mean when you say these are single-celled. So say these are single-celled organisms all right, so they are single-celled organisms. So the best example you can give is a bacteria. So a bacteria, so for us even to talk about the characters, let me give you first the structure so that we'll be getting all the characters from just the basic structure of a bacteria. All right, so I'll draw a bacteria here. All right, if I'm not mistaken, I don't know if I still remember a bacteria, but I'll draw it. Either guys, I'm not good at drawing, so an apology for that. Okay, so we'll say this is a bacteria. Okay, so we'll say this is what we call now a flagellum. Let me not do this. So prokaryotes, they are single-celled organisms. All right, so that is the a tail to say. All right, so that's a tail. We'll say that is a tail. So there's another layer inside there. So there are 3 layers of this guy. So it has got 3 layers. All right, so master the layers. By the way, it's not supposed to be like it's an animal, so it's not supposed to be regular like that. You just draw the way you wish to draw. So yeah, you remember those characters to say a plant cell is regular, but for these guys, animals. Okay, so that is it. Then let me draw something inside there. Okay, okay, so once we master the characters or just these parts, then we'll be able to do what? We'll be able to okay, so that's just like a structure which looks like a hand or like a code thing there, like a blue one. So the other thing that we can say is now let's label the parts. So these parts, once we know how to label them, then we'll be able to know that this is the part. We are going to extract the characters from the same structure. All right, so let's master this structure. So this tail that you are able to see, it's called a tail, right? Not really a tail, we'll call it a flagellum or flagella, just a flagella, but let me call it flagellum. All right, okay, so we know the function. Or let me just put a function for flagellum. So the flagellum will be used for movement. So it's the one which makes the swimming process, if you were to say like swimming, to be easy. So for movement. All right, so a flagellum will enable this guy to move from one place to another. So for movement. All right, so this thing, the yellow part, you are able to see there. By the way, when you are labeling here, we just label like this. We don't label putting the arrow direction, we just label like that. So in case you are labeling to say like this, we don't do that here. All right, so here what we do is we just put a line, then from there you name the part. So this part, with the one which is just outside, we call it a capsule. All right, so it's called a capsule. All right, so this capsule is more like adherence material that attach to the host. So it's the one which is more like it has magnet. Once it attaches there to your skin or any cell, you say a cell or a skin of your cell, then this one will enable it to be, what's this, to be attached like effectively and without even wanting to leave that position. All right, so it's a material that do what, it's more like a adherence material that attaches. So this one used to attach to the host. Okay, so a host is something that is now affected. That's a host. When you host something, it means you are carrying something. So a host is the one which will carry this. So a capsule will be used to attach there. It has even, like, we can say, some hooks, if we were to describe like that. So the inside now, the inside part that you are able to see, the blue part there, this one is called this is now a cell. It has a cell, by the way. It has some character of what, a plant cell, whereby we can see a cell. So this is a cell. All right, so since that is a cell, what is the function of a cell? Anyone to give me a function of a cell? All right, we know it's some kind of protection. Now in this case, we have also a capsule that protect this guy. Okay, so a capsule will be there to protect, even a cell will be there to protect the lead part. Let me label, in fact, the lead part there. Okay, so the lead part that you are able to see there, it's what we call the plasma membrane now. So it's a plasma membrane. So we'll say the cell will protect the plasma membrane. Simple. So these things, this green thing, you're able to see inside there. Okay, so those green thing you're able to see out inside there, they are what we call ribosomes. So ribosomes. So these ribosomes will be there to synthesize a certain molecule, that is protein. Okay, so ribosomes. So you guys should master this structure. It's very important. You can be asked to say, when you are giving the characters of, you can still label, you give the structure and the label, you'll be awarded for that. You'll be given some marks. All right, so this ribosome is used to synthesize protein. Synthesize, it's more like making, right? So yeah, synthesize proteins. Okay, so it will be used to synthesize proteins like that. Then the other part that we can talk of is this part here. Let me label it from this side. This blue one, this blue part, you're able to see this gamma thing which looks like a hand. This is what we call a mesosome. All right, so you see a mesosome. So what is its law? A mesosome, what is its function? So a mesosome will be there to produce, or that's where now energy will be formed. So production of ATP. ATP is the energy available for cells. So the energy that is about to be used is what we call adenosine triphosphate. All right, that is ATP. So production of, so say this one will be in charge of production Okay, making of energy, production of, I'll call it ATP. We know what ATP is the energy available, readily available for cells. So that is ATP. There might be ADP and other forms of energy, but for ATP is the energy readily available for cells to be used. All right, so it will be there to synthesize or to make that energy. So the energy of these prokaryotes will be found or will be formed from mesosome parts. All right, so now we can draw now the characters. Okay, so we can draw the characters now. So say characteristics with the difference to the diagram. So characteristics. So what are these characters? We'll say they are single-celled organism, of which we have already talked about. So we can say it's just a single cell. This one, a bacteria is a cell. All right. So it has no nucleus. So I've forgotten one point here. There's this guy here that I've just forgotten. All right. So it was supposed to be a nucleus. Now in this case, it's not really a nucleus because it's not round like that. It's more like unfinished made nucleus. So we'll call this as a what? A nucleoid. All right. Instead of nucleus, we'll call it a nucleoid. All right. So a nucleoid, from the word something, a nucleus, okay, a coid something is something which is just, rounded like that. When you say coring is something that is like this, is something that is cold. So the word cold means something that is just rotating itself, and that's what we call a coring material. So a nucleoid it will be there instead of a nucleus from these others, which are other cells. All right, so we say it has no nucleus. Instead it has a nucleus. What it has, so say it doesn't have a nucleus. All right. Instead it has what? It has a nucleoid. All right. So that is the nucleus of this guy. Then the other thing that we can talk of is it has a cell wall. So we are getting from this now. It has a cell wall. Other cells don't have a cell wall. All right. So apart besides this, this guy, we can see that it has a capsule which protects the cell, and also it is used for attachment. So these are the just characters. It has what? So here it has a cell. Here it has what? A capsule. All right. So this capsule will be used for protection of the cell against damage, and also just for the attachment to the host cell. So a capsule will be used in those cases. All right. So the other thing that we can say, these guys, for them to replicate, they don't just replicate like reproduction, normal reproduction, sexual reproduction. No, they go what we call binary fission. All right. So they undergo binary fission. All right. So a binary fission is a situation whereby it just develop, a bad something like that. We'll talk about binary fission processes. Okay, we'll talk about it. We'll see how it is done, how a mother forms a baby. All right, so a binary fission, so we call it binary fission. So the other character we can talk of, it has a flagellum which allow the movement, right? We have talked about this, so we are just extracting from the diagram. So say it has a what? It has a flagellum. You are telling them now all the prokaryotes have flagellum, all right? So all the prokaryotes now have a flagellum. Okay, so it has a flagellum which allow the movement using the energy from ATP. So it will be using this energy made from the from this guy. So it will be using that energy made from mesosomes to undergo or to perform movement, or just other attachment of which we have already talked about. All right. So it has a flagellum used for movement. Using the ATP energy. We have talked about ATP energy. We all know what ATP energy is as for now. So the other thing that we need to know is there's this what we call a presence of peptoglycan. So there's a layer between a cell and a capsule there. All right. So between the cell wall and cell membrane there's a layer between a cell wall and a cell membrane. There's a layer there, all right, of which we can't really see there. There's a layer that we call a peptoglycan. So this can contain peptoglycan. So say a layer between the cell, between the cell wall. So say between the cell and cell membrane, of which is a plasma membrane, cell membrane. So between the cell and the cell membrane, there's a layer that we call a peptoglycan. So a peptoglycan is a layer that we find. So there is what we call peptoglycan. All right. Peptidoglycan. So this peptidoglycan, you can talk of peptide, simply means protein. So we can say pepto, this just ending from here. This is what we call pepto. So this will call proteins. All right. Relating to protein is a peptide, something like peptide bond. Then glycan relating to glucose, of which is now we can call it carbohydrates. All right. So it is made up of proteins and carbohydrate. All right. So this peptoglycan is made up of glucose and carbohydrates and the protein. Or peptide simply means protein. This word peptide simply means protein. All right. Then glycan simply means carbohydrates. All right, which is only found in bacteria and not in archaea. So in archaea, I've given two examples of these guys. We said bacteria and the bacteria, all right, and archaea. All right. So these are the examples of prokaryotes. But for this case, for this region or a layer, this is a layer. It's called a layer. All right. It's a layer on its own. So this layer is found between the cell wall and plasma membrane, which is cell membrane. So it will be there to determine the type of bacteria it will enable us to check what bacteria are we dealing, let's say, in this infection. So the presence of that will enable us even the treatment itself. So it is only found in bacteria, not in archaea. So their cell walls is made up of this layer, okay, these bacteria. So some bacteria can be gram positive or gram negative. So a gram negative, it will be a procedure that will be used to check whether the bacteria we are dealing, in short, it will be there to enable us to know, knowing the bacteria that we are working on it. Let's say this bacteria, let me go on the next page. Okay, so the bacteria, so we'll say now the bacteria that, let's say this is a person injures, there's a sore here or a wound. So this wound is keeping on, like it's growing each and every day. It's more like it's a cancer, or you can say a sore. So this one, there are bacteria enabling to like to grow, because every day it's more like it has dead. So bacteria are now developing there. So we want to see the type of bacteria, and so that we can treat. There are bacteria that don't have this, or which has thin of this peptoglycan. So what we do, we conduct an experiment there, a procedure that what we call gram strain. All right, so a gram strain will be performed on that infected area. So perform that this process, okay, of which we'll talk it in the next video. Okay, so that is about it. So a layer will be used for that case. So don't even get confused. A layer will be used for that case. So some bacteria will contain a negative layer, which is negative grams 10. Some will contain a positive grams 10. So we are talking about some bacteria who have, when we say a gram positive like that, meaning it has a thick what? Thick thick layer, of which the layer we are talking about is peptidoglycan. All right. So a gram negative now, it will contain a thin, all right, a thin peptidoglycan like that. So for once we know the type of bacteria we are dealing with, then it will be very easy for us to know the treatment. Okay, for the treatment, all right. Then apart from the treatment, we also know the type of bacteria we are working on it. Let's say we are performing a research, we'll be able to know, to say, oh, the bacteria that we are working on is this type of bacteria. All right. So now we'll talk about, we can't just be studying something without the importance and also or just the benefit. Each and everything has the importance and also the disadvantage part. All right, so now we need to appreciate these guys. Of course, they might bring other diseases, but let's just appreciate other laws. They play some important laws in our environment. All right, so economic, you'll say economic. Oh. We can't just be studying something without the importance. Why are we interested when we can't see anything? No, there must be some important part. So that's what we call now economic importance of prokaryotes. All right. Okay. So these are the things that now I want to talk about. These guys will be important in that they'll be there to be used in medicine, e.g. the medicine that we know which lower glucose level in our blood. That's what we call insulin. So production of medicine, so we'll be using bacteria to make medicine. All right, production of medicine like that. E.g. insulin, a well-known hormone. All right, so we'll be using also these guys in decomposition, decomposing matters. All right, so it is because of this their presence we can have things that, or decompose things faster. Okay, so even these organisms that dies, they will be, because of the presence of bacteria, they decompose faster and will be able to recover things that we want from that. So it will be also used in beer production. All right, so the other thing that we can use these guys is in the maintenance of carbon cycle. So they'll be there to enable or to make the carbon cycle, the process to continue. So maintain carbon cycle will be maintained so that it can return to the atmosphere through the bacteria. All right, through the bacteria like that. Okay, so that is it. To maintain carbon cycle. Okay, these bacteria will be using this study. So these are the things we need to to bear in mind as we study prokaryote. So that's about prokaryotes. We have talked about the examples of which it is archaea and also bacteria. We have talked about just the structure characters, of which we drew some characters from the basic structure of the bacteria. So the bacteria structure will give us more information about prokaryotes. So the next topic we'll talk about now the grand strain procedure. So don't miss the next lesson, please. When you want to contact us, you want lessons for engineering mathematics, BI 110, MA 110, PH 110, and also 260, that's organic chemistry. So you'll be able to reach us. So just contact us. See you in the next video.